its journey from the 18th, and that is the long lasting journey, and that is coming in a real phase. Now we are coming to the next question, that why bioinformatics should be studied, that what are the basic applications of the bioinformatics. Because basically when we are talking about the bioinformatics, initially people thought that whatever the persons are involved in the biology, that should be included in the bioinformatics. But when we are taking a role of a real biologist, then they are finding that the biology people are not understanding the computer well. So they will feel that the computer science people should come. But when we are observing that this is a multidisciplinary science and we should have an equal role of so many people together, so for that the bioinformatics application, we have to go through the different areas where it is applicable. Why we should enter in the bioinformatics? So that way, suppose comparing, then bioinformatics is a science of a smart people, and that is making your life very easy. If you are comparing the normal experimental biology, then you should go through the long lasting lab protocols. You have a lot of restriction when you are entering inside the lab. You have to wear a specific kind of the apron, and lot many restrictions are there. But when you are entering in the world of the bioinformatics, it looks something colorful like this only. There is only limitation is that you should be computer savvy and you should be a friend of computer. Then you can find out lot many things out of that. And even that is a more smarter and speedy way of performing the things. Now how the bioinformatics has developed, that has given us a new dimension in the entire biology. So this we can compare like that when initially people have started working on the field, then we are having a limited tool, and with the limited tool only we can construct a small hut, and we got the lot of superstitions and lot of other things whatever we had in the 17th century and the 18th century. But when we have entered in the 19th and 20th century, we got lot of modern tools, and this has changed the entire scenario. We got the modern house, we got the modern development, and along with that only lot of biological equipment has also came, and due to that our thinking about the biology, that is also getting changed, and that is entered in the era of the omics. And right today now we are in an era of the omics. The omics is a holistic way of studying the entire thing. Right now you can observe that common terminology which is normally utilized for the genomics, proteomics, transcriptomics, metabolomics, lot of omics are coming. But this till today whatever is coming, that is also a tip of iceberg. Lot of other science are also coming today. So it is not a thing of wonder that after a few years you can find out that the normal degree like the MSc Microbiology, Botany and Zoology, that will have the new titles like the MSc in the Genomics, MSc in the Proteomics, and lot many other science are coming in the field. So that way we are entering in the slowly the world of the omics. And the world of the omics is basically having a fundamental role of the bioinformatics within that. Suppose you want to understand this omics world, then you have to go in a systematic manner. We had the initial law of the Darwin, and after the development of the Darwin slowly we had the lot of information about the DNA structure and sequence. After that we have stored all the information inside the computer, and when we have merged the lot of bioinformatics fundamentals whatever we have seen earlier, we entered in the world of the omics. Suppose you are observing this omics world, then that is a matrix like structure. You can observe that study of the entire genome, that is normally taking in a part in a living form, then that is coming as a genome. Suppose we are taking the entire transcript, then that is normally known as the transcriptome. Similar to that, proteome, interactome, functionome and texotome is coming. But when we are merging this first row of the information with the sequence, structure, expression, pathway, regulation, network and all other information kind of the information along with that, then we are coming to the second generation, and that converts the genome into the genomics, transcriptome into the transcriptomics, proteome into the proteomics, and like so many others. And whenever you are converting the normal science into the omics, the fundamental role of the bioinformatics is coming always. Why the bioinformatics having the vast application nowadays? Because now we are observing that now we had the lot of techniques by which even you can have the entire sequence of the genome within a few days. Now even in Gujarat also we can have a facility by which you can have the complete genome of the microbes within few days only. And that way throughout the world is generating lot of data, and we are not having that much speed even to analyze all this data with our normal human brain power. And for that we are in need of the bioinformatics that will help us. And our condition is something like that whatever data is coming like this, that is having a flow of this much, and our need would be quite something less than that. So we require some kind of the mechanism by which we can reduce the entire flow and we can extract the things whatever is required for our thing. Suppose we are taking the fundamental application of the bioinformatics in a different part, then we can see that the first application it is having in the science of the genomics. So genomics is the study of an organism, entire genome, and genome sequencing of bacteria, viruses, animal, plants is done. After getting the sequences only, immediately people are going for the comparative genomics, annotation and mapping, and this includes a process of gene discovery. So basically this is a process like this, that suppose you are running the entire sequencing of the HIV, then you can get the physical map of the HIV. That would be merely a sequence of A, T, G, C and other. But from this simple sequence of the ATGC, you will be not finding any meaningful information. So to convert such kind of the information into a meaningful information which can be utilized by the different biologists, you should have some technique by which we can convert the entire information in this manner. When we are observing the same map, in this manner you can find out that the entire thing is getting annotated. We can easily find out that first sequence is coming, that is for the gig. After that we can have the sequence of the pole, and after that we can have the sequence of the envelope. So like that only we can find out that from which place one gene is getting started and at which place the one gene is getting ended. And when we are having the such kind of the information, we can get the lot of information which can be utilized by the biologist. It can be utilized by the doctor to create a new kind of the drug. It can be utilized by the different kind of the biologists to find out the evolutionary relationship. It can be utilized by the microbiologists to find out the epidemiological outbreaks and lot many other applications. So that way the genomics that will convert the raw information into the meaningful information for the biologist. Similar to that, the second important science is coming, that is normally known as the proteomics. So this proteomics that stands for the large scale study of the protein, particularly their structure and function. Suppose we are grossly examining the entire things of the proteomics, then the proteomics something like this. Suppose we are comparing the sequencing technology which is available today for the DNA, then that is quite rapid. Within few days only you can get the full sequence of the DNA. But suppose you are comparing the same thing with the protein, then even till today we are finding that the sequence of the protein is somewhat difficult task. And suppose you want to completely sequence the entire proteome, then that becomes a quite difficult task for us. So to take a shortcut only, normally we are going through the process which is normally known as the 2D gel electrophoresis. This 2D gel electrophoresis, that is a simple process by which you can separate all the proteins which are present in the cell. Normally in the E. coli we are having the different 2000 protein at a time that can be separated by this gel technique in a smaller spot. Each of these mode can be cut, and after that can be digested with the different kind of the proteolytic enzyme. When it is getting digested, that will creating a short fragment of the proteins, and after that the short fragment of the proteins that can be analyzed with the help of the Marlitov, and that will just give us the molecular weight or the size of all of this fragment. And after this we can compare this fragment size with the database which is available in the biological database, and by comparing the size fraction of this we can find out that your protein is having the similarity with which kind of the protein in the nature. So that way this proteomics that is shorten our entire time. That suppose we are going for the sequencing of the protein, then that is beyond reach for us right now. Suppose I want to find out that which is the genes which is responsible for the cancer, then I can have a simple protocol of taking the cancerous cell and taking the normal cell, and after that I can compare the 2D gel image of both of them, and I can pinpoint a specific spot which is remain absent in the normal person and which is present in the cancerous person. And after that when I'm going through the protocol, whatever we have just observed, we can find out a sequence of that protein which is matching in the database, and immediately we can find out a desired gene for that. And when we are finding the gene, then we can got the lot of opportunities to treat them in a proper way. So that are the fundamental application of the bioinformatics. Similar to that, even the bioinformatics is having the lot of application in the metagenomics. The metagenomics is a study of metagenome, that is a genetic material that is recovered directly from the environmental sample. This metagenomics is having the vast application typically in the field of the microbiology, because as we all know that less than 1% microorganisms can be cultured, and that way we are having the lot of other microorganisms in a nature that is not sequenced, that can be not cultivated due to the inability of cultivating them in the lab condition. So due to that only we are not having any ability to use the genetic information which is existing with them. With the help of the metagenomics, now it is possible to reach to such kind of the uncultivable gene. How it can be done? It is a very simple process when we are going through the entire metagenome. Metagenome is a process by which we can extract the all DNA from the microbial community in a sample environment. We are normally having the extreme environment. For example, in this figure only some deep sea environment is shown. And whatever the organism is surviving under such condition, to grow them in a lab condition, it is a very difficult task, because we cannot mimic the same kind of the environment inside the lab condition. But if you are using the metagenomics, then we can extract the total genomic information from them. Whatever the metagenome is isolated, that metagenome can be analyzed by two manner. In the first manner, we are having the sequence-based metagenomics. In that, normally with the help of the next generation sequencing, the entire metagenome is sequenced, and after that, with the help of the different bioinformatics tool like the MGRAST and so many other, the gene is getting identified that can be compared with the metabolic pathway and other communities. So that way, without cultivating them only, we can get the lot of information from this. Similar to that, even the functional based metagenomics is possible, that whatever the metagenome is extracted, that metagenomics is normally cloned in the different kind of the vectors like the cosmid, even plasmid, and some kind of the back vectors and other, and after that we can functionally screen them for the important function. So even though we are not directly cloning them, but we are cloning the gene which is required for our purpose only. So that way we can have a snapshot of the required gene from the entire process. And to carry out this entire metagenomic exploration, we require the bioinformatics application always with that. Similar to that, even the bioinformatics is having the vast application in the field of the drug and vaccine design. Basically, drug is coming from the natural product, plant, fungi, bacteria and chemicals, and the screening process of the drug, it is becoming a quite normal process in the earlier time, and that is normally known as the blind searching. Blind searching means we are not knowing anything about the disease what we want to cure, we are not knowing anything about the activity, but normally we are screening depending upon the method of the trial and error. As we all know that the penicillin was the first antibiotic that was discovered, so that was discovered by the simple process of a normal screening only, and that was also a chance phenomena. Similar to that, in the earlier time, people used to go through the normal protocol. When we are observing the normal protocol of the drug discovery, then that is going through this entire process. Always in the drug discovery, we are having the first step of identifying the disease. After identifying the disease only, we have to identify the protein which is responsible for that. After getting the relevant protein, normally we are going for the searching of a drug which is effective for that, and after getting some of the good drug list, then it is normally entered in the preclinical testing. The preclinical testing that includes the testing on the animals, and if your drug is finally successful in this stage, that is normally entered into the laboratory scale scale-up and even industrial scale formulation, and after that people are giving this everything to the human clinical trial, and from the human clinical trial, depending upon the nature of the drug and problem, it will take around 2 to 10 years to get completed, and then finally we can have the FDA approval, and from that only you can find out the entire drug which is coming out. So that way, suppose you are comparing the entire drug discovery pipeline, then that is taking 10 to 15 years, and even it will take a lot of money to get through the new drug out of that. Now with help of the different kind of the bioinformatics technique, whatever we have studied right now, we can accelerate the entire path. Suppose I want to find out a specific protein, or suppose I want to get the information about some new disease, then with the help of the genomics, proteomics and biopharma, I can find out that whichever receptor which is responsible for that disease, and from that only I can get the lot of information related with that. After that, the second part is coming, that is normally done inside the in vitro condition with the high throughput screening, that is also having the capacity to screen around 1 lakh compound per day. But now with the help of the computer, we can enter in a phase where you can go for the virtual screening. And when we are going for the virtual screening, in one day only you can screen the millions of the compound inside the computer. And at the same time, you can also observe that whatever the process of formulation, that can be also omitted at this stage. First of all, you can generate the compound inside the computer in the in silico environment in a virtual manner. And in a virtual manner only, with the sequences which we are getting from the PDB protein data bank, we can run the screening exercise. We can go for the docking studies and other, and by which we can list out few chemicals which are quite effective, and only after that, that few chemicals can be synthesized inside the lab condition. So that way, that is reducing our efforts, and at the same time our speed is also getting increased for the virtual screening. Now after that, when we are going for the combination chemistry of the entire drug, then we can find out that some of the drugs are quite good, but if you are changing some of the portion of that drug, then that will become more effective. Even in market also, it will happen that initially we had the penicillin, but after few years we are getting that the microorganisms are getting resistance towards the penicillin. Then we have started using the ampicillin. If the organisms are getting resistance toward the ampicillin, then we are finding the new derivatives like this. So that way we can alter the basic drug molecules, and according to that only we can find out the new drugs out of that also. So that is a part of the combinational chemistry, and even parallel to the bioinformatics, science of the chemoinformatics is also developing in a parallel fashion. Now after getting the good drug out of the second phase, normally we are entering in the preclinical testing. In the earlier time, the preclinical testing was a must, and that is also quite cruel in nature. We are sacrificing lot of animal for our own purpose of a lab. But now with help of the bioinformatics, you can have the in silico model, which is generated depending upon the neural network softwares, and that can predict the entire activity inside the animals. So that way, without using or without injecting drug or without sacrificing the animal, we can get the lot of information about the different bioinformatics application. So that way, that is reducing our entire pain, and whatever the duration we have observed, that is coming from the 0 to 15 years, that can be reduced in some way, that you can find out the drug in a very short time with help of the so many reduced money also. Suppose today we are observing the different kind of the drugs which are available in the market, then more or less for lot of drug, this kind of the bioinformatics approach is taken. Suppose you are taking a blockbuster drug of the Viagra, then even the invention of the Viagra also, the different kind of the computer-aided technique was utilized. So similar to that, even till today, people are utilizing this combination technology for searching the new drug for the anti-HIV, anti-cancer, anti-malaria, and lot many things are there. Similar to that, suppose you are taking a case study of the bioinformatics, that how the bioinformatics practically can be applicable. So we can just have a simple example that now we are also having the emerging of the new diseases. For example, before few years, we had the outbreak of the SARS. When the SARS outbreak came, immediately we got the lot of case in the market. We got that lot of patients are getting died every day, and even the disease is getting spread like anything. So at that time, we are not having that time to go through the traditional process, and we can find out the new drug out of that. Suppose you are following that old process, then we can find out that lot of people getting died of due to the emergency outbreak of the SARS. Suppose we are taking the bioinformatics along with the SARS, then what is happened at that case, that whenever the outbreak of SARS was, that immediately people have came in the action, people have started sequencing the entire genome of the SARS, whatever the viruses are there, for that the full information is taken from the sequence, and after getting that sequential information, people have gone from the different kind of the algorithm, and immediately they find out the different kind of the solution out of that. So when we are getting the partial sequence of the SARS, we can go for the various genomics, proteomics, and all other kind of the exercise whatever we have seen earlier, and that will accelerate our entire efforts to find out effective drugs against the SARS or any other disease in a practical application. So that way, whenever we are having the emergency and whenever we are having the lack of time, the bioinformatics is coming an important tool for us. Now the next question is coming, that who is bioinformatician? That when we are talking about the bioinformatics, we are telling that that is a multidisciplinary science. We are taking the help of the microbiologist, we are taking the help of plant science, we are taking the help of animal science, we are also taking the help of the mathematician, computer softwares, and lot many people together. So that way we are not having a single person who will have all the information about the bioinformatics. So the bioinformatics is something like this. It is like a Ravana. The Ravana will